The first week of development contains the following stages. Ovulation. So the secondary oocyte ovulates out of the ovary. Fertilization. The definite egg gets fertilized by the sperm. Cleavage. After fertilization, the formation of zygote takes place, and this zygote goes through the process of mitotic cell division multiple times, and that's what we call cleavage. Morula formation, that is when the formation of morula takes place, and blastocyst formation, that is when the formation of blastocyst takes place. So first there's ovulation, then fertilization, then cleavage, then morula formation, and finally the formation of blastocyst. So the first week of development ends with the formation of blastocyst. Out of all these stages, we have already dealt with ovulation, and I already have a video posted on fertilization. So in this particular video, we are going to complete first week of development from cleavage till the formation of blastocyst. First is cleavage. After the definite egg gets fertilized by the sperm, the formation of zygote takes place. The zygote itself is a single cell stage. The zygote is still surrounded by a glycoproteinous layer called zona pellucida. The zygote goes through mitotic cell division. Here, the zygote goes through first mitotic cell division called the first cleavage, forming the two cell stage 1, 2. The two cell stage goes through second cleavage or second mitotic cell division, forming four cell stage 1, 2, 3, 4. The four cell stage goes through third mitotic division to form 8 cell stage. The cells that are formed from mitotic cell division are called blastomeres. The 8 cell stage again goes through another cleavage and finally forms a 16 cell stage. This 16 cell stage looks like a mulberry, and that's why we call it the morula. So this is where the formation of morula takes place. The cells of the morula rearrange themselves to form two different masses of cell. The mass of the cell that is present at the middle, called the inner cell mass, and the mass of the cell surrounding the inner cell mass, called the outer cell mass. After this begins the process of formation of blastocyst. Next, blastocyst formation. So we have reached till this particular structure. It contains the inner cell mass surrounded by the outer cell mass, and this is still present inside zona pellucida. This particular structure reaches your uterus, and the uterus contains endometrial fluid. The endometrial fluid enters inside or penetrates through zona pellucida. As a result, a large cavity is created which is filled with endometrial fluid, and this cavity is called blastocele. The reason being, this is a cavity which is present in blastocyst, and this right here is the blastocyst. During the formation of blastocyst, the formation of a cavity named blastocele takes place. As a result, the inner cell mass gets pushed towards one particular end. Now the inner cell mass and the outer cell mass are no longer called so. The inner cell mass is now called the embryoblast because it forms the future embryo, and the outer cell mass is now called trophoblast and it forms parts of placenta. The pole of the blastocyst towards the embryoblast is the embryonic pole, and the pole opposite to the embryonic pole is the abembryonic pole. This whole structure right here is called the blastocyst. And the blastocyst, as you can see, is still present inside zona pellucida. However, the zona pellucida slowly ruptures and degenerates, and the blastocyst comes out of the zona pellucida, and this is called hatching of blastocyst. Basically, blastocyst hatches out of the outer zona pellucida membrane. This right here is the hatched blastocyst, which contains the outer layer of cells called trophoblast, which forms parts of placenta. The inner cell mass present at one particular pole called embryoblast. Here's the embryonic pole and here's the abembryonic pole, and this large cavity filled with endometrial fluid is the blastocell. The blastocyst then starts the process of implantation, where it gets implanted to the wall of the uterus, from the embryonic pole and not from the abembryonic pole, from the embryonic pole. The rest is the second week of development. So this is it for first week of development. So the first week of development includes the ovulation of secondary oocyte, the fertilization of definite egg by sperm for the formation of zygote, the mitotic cell division of zygote, the formation of morula, and then finally the formation of blastocyst. The first week of development ends with the formation of blastocyst. After fertilization has taken place, the formation of zygote takes place, which goes through multiple mitotic cell division to form morula, which is a 16 cell stage. It is called morula because it looks like a mulberry. The cells of the morula rearrange themselves to form the inner cell mass and the outer cell mass. The inner cell mass in future forms the embryoblast, and the outer cell mass in future forms the trophoblast. Blastocyst formation starts from this particular structure, which is present inside the uterine cavity. The uterine cavity contains endometrial fluid, which enters inside this structure and forms a large cavity filled with endometrial fluid called blastocell. As a result, the inner cell mass gets pushed towards one particular pole and is now called the embryoblast because it forms the future embryo. The embryoblast is surrounded by trophoblast, which forms part of placenta. The pole towards the embryoblast is the embryonic pole, and the pole right opposite to it is the abembryonic pole. This structure right here is the blastocyst, which is still present inside zona pellucida. Finally, the blastocyst hatches out of the zona pellucida and forms a structure like this. This structure or blastocyst gets implanted on the wall of the uterus at the embryonic pole, and the beginning of the second week of development starts.